Hi folks, welcome back to the experimental design and analysis section of the core skills modules, and we're making good progress. We're on lecture 8 of 10 about the sort of theory of experimental design and analysis, and today I want to talk to you about a thing called ANOVA, which stands for analysis of variance, and it's an absolutely key technique, statistical technique, for evaluating biological differences. So as a reminder, we're talking about differences. My pet topic, my favourite research topic is mimicry in the natural world. Here are two fungi on screen. One of them is deadly. If you eat it, you die. The other one looks a lot like it, but actually is very palatable and good to eat. So you find mimicry all over the place. Not hugely relevant to stats, but I like it. So let's, oh yeah, there you go. There's the tasty one and the deadly one. Let's have a look at the outline for today's lecture. It's going to be in 3 parts as usual. We're going to first of all look at basically the sort of principles of analysis of variance. So we're going to look at how different experimental designs lead to rather more complicated difference tests or difference comparisons than we've seen previously. So I'm going to put this in the context of what we looked at so far and then how life gets more complicated, specifically how we have potentially multiple levels instead of just 2, which is what we have so far, and potentially more than one factor. At the moment we've only looked at examples with 1 factor. At the moment those words will mean nothing to you. It will become clear in a moment. Then we're going to look at the principles of analysis of variance in particular, so how it works, and your objective for the lecture really is to try to understand those principles. And in order to understand them better, we're going to look at a worked example in the middle part of the lecture. So a detailed example about bird morphology. And then towards the end, we're going to touch on a related topic called post hoc testing. A post hoc test is something that you might do after analysis of variance, and I'll explain why and what it is. And then finally, we're going to look, as with all parametric statistical tests, at the assumptions. So just like a t-test or a Pearson's correlation, ANOVA makes a bunch of assumptions about the data. We need to think about those before we press ahead with the analysis, and if we can't meet those assumptions, we'll look at a non-parametric alternative, and actually we'll look at that next week. Okay, so part 1 is to look at the basic principles of analysis of variance. So thus far on the course, we've looked at differences in a number of contexts, but we've focused mostly on situations in which there is only one factor, and that factor has 2 levels. So those 2 words, factor and level, are important pieces of jargon. Let's explain what they mean with an example. So if you remember back a few lectures, we looked initially, when we were thinking about t-tests, at a comparison between sperm that we got from sperm that had been treated with calcineurin and sperm that hadn't. I'm just trying to think, was it not calcineurin inhibitors? I think my slide might be wrong there, it doesn't matter. Anyway, there was a drug treatment and there was a control, and we were interested in whether it might affect sperm swimming speed, potentially with some useful side effects in terms of male contraception. But the crucial thing is, we had just 2 treatments, 2 levels as we call them, the control and the drug treatment, within a single factor. And the factor here was, well, we might broadly call it the treatment, control versus drug. In other words, there was one variable with 2 categories. The variable in this kind of context is called a factor, and the categories are called levels. So another example we looked at was these snails that are a bit weird, and they can choose to have sex with themselves and reproduce asexually, effectively, or parthenogenetically, rather than finding a sexual partner. And we looked at whether they do that more often in the presence of a threat from a predator. So here, the thing that we're manipulating in the experiment, the variable of interest, is the presence of the predator cue, and it's either present or it's absent. So we've got 2 levels, present and absent, in a single factor, which is whether or not the predator cue is there. And then finally, last week, we looked at some examples of chi-squared tests, and we focused on, for example, this factor for blue tits in an experiment, whether or not females were mating with low or high quality males, depending on their plumage, which depended on whether they'd been covered in sunscreen. Again, we had a single factor, which was the presence or absence of the sunscreen, and 2 levels, either it's there or it's not. So far so simple. But actually the real world is more complicated than that, and lots of experimental situations require us to consider multiple levels within a factor, and indeed multiple factors simultaneously. So on screen here you've got an example of what I mean. First of all, a single factor with more than 2 levels. So what you can see on screen is 4 life stages of a ladybird, from egg through to adult. So it goes egg, larva, pupa, adult. And in the particular experiment I had in mind here, we might be looking at the concentration of a chemical called pyrazine, which is shown on screen. And that pyrazine is actually a very nasty, bitter tasting substance which predators really don't like. And that's one of the reasons why ladybirds are black and red. They're actually advertising this, giving a warning signal, telling a potential predator that they contain this nasty chemical, which will actually potentially make birds vomit if they try to attack these beetles. So we might be interested in whether pyrazine is present in all 4 stages of the development of the ladybird, or if perhaps it only develops in the adult, or at some point in between. So we could measure, using appropriate biochemistry, the levels of pyrazine in the different life stages of the ladybird. I mention this because actually this is some work, or similar work, has been going on in the school. You might have met Ian Mellor, who works on various neurotoxins, and he's been interested in how pyrazine interacts with neurotransmitters, and also in the context of ladybirds. So what we've got here is one factor with 4 levels. The 4 levels are the 4 developmental stages. The factor is developmental stage. But you could easily imagine experiments in which you were interested in the levels of pyrazine not just in one species, but in several. So on screen here I've imagined that we're looking at 2 different species of ladybird and all 4 life stages of both species. So we end up with a kind of 4x2 design. And what we've got here is 2 factors. We've got life stage and species. One of the factors, life stage, has 4 levels, and the other factor, species, has 2 levels. So hopefully you got your neck around this factors versus levels business. The factors are the categorical variables that we're interested in, how they affect some response variable, and the levels are actually the numbers of categories that we have in each of those variables. Okay, so when you get to a situation like this with lots of different levels and multiple factors, you obviously can't perform a straightforward t-test, because a t-test assumes you've just got 2 levels in one factor. So you are potentially, I'm just getting called on another line as it were, so I'm just going to cancel that. You're potentially going to struggle, you are going to struggle to use a t-test. You could consider making pairwise comparisons between individual life stages, so we could compare the egg with the larva, or we could compare just the adults of one species with the adults of another species. But you'd rapidly end up with a whole load of tests, and there would be various problems with that approach, which I'll go into later. So the magical solution is actually a parametric statistical test called analysis of variance that allows you to evaluate any number of factors and or levels simultaneously. So it's a really powerful statistical test, and it is very flexible because we can use it for lots of different situations. So that's what we're going to look at in today's lecture. So if we just focus for the time being on a single factor, imagine a single factor that has more than 2 levels. On a frequency histogram, as I've sort of shown you in previous examples, it might look something like this. We've got 4 different samples represented by 4 different colours. So we've measured a single response variable, like the concentration of pyrazine, and we've got 4 different groups of data points, each representing a different life stage. And if we have a large enough sample and we plot them on a frequency histogram, often we're going to see a kind of bell-shaped distribution, and we might see different distributions for the different life stages. And what we want to know is, are the individual levels of the factor, or the groups in the experiment, or the treatments in the experiment, are they significantly different? You'll notice there's lots of sort of different words we could use here to describe these groups, and that's one of the frustrating things about stats, is that different people use different words. So I think we just have to kind of live with that. But hopefully it's intuitive to you that we can have lots of different types of experiments with multiple groups like this on a single factor. So there's a single variable of interest here, which is life stage, or in my example it was life stage. So we want to know, are they different, or do they in fact all come from the same underlying population with the same mean, and any differences that we see between these different samples are just random chance? This is the sort of basic principle of our statistical hypothesis testing framework. We're always asking, is what we're seeing likely to have happened by chance in a world in which nothing much interesting is going on? In other words, the null hypothesis is true, there are no differences between the groups. Or are we seeing something more interesting than that? Are we seeing a genuine significant difference between groups? And in the case of ANOVA, it's important to note some jargon again, that the mean of all of the data put together, so if we pooled all of these 4 samples together, the mean of those data would be called the grand mean. And that's a term we're going to use later on in order to do analysis of variance. And so essentially the question we're asking with analysis of variance is, do all of the data come from a single population with a mean equal to the grand mean, or estimated by the grand mean, or instead are we looking at 4 separate samples representing 4 separate underlying populations, because in the case of our ladybirds there really are significant differences in pyrazine levels according to life stage. So apologies for me drinking my tea, but it's late in the afternoon, I need my caffeine. So what we've got really is 2 sorts of variation to think about in order to answer this question. We've got variation within a group and variation among groups. So for each of the groups or samples or treatments that we've got, there's going to be variation naturally within that group. So if I take eggs of ladybirds and I measure pyrazine concentrations in each one, of course I'm going to see variation. And that variation is visualized in a frequency histogram by the spread around the mean for that particular group or sample. So here I've indicated that by x bar 1, that's the mean for the first sample. And similarly, we're going to have a mean for each of the groups and then some spread around it, some variation around it. But in addition to that, we've got a different source of variation, which is the variation among the means. So each of the 4 sample means is different from the grand mean. And the grand mean here is indicated by x bar, double bar on top. And so each mean is a little bit different, so we get an extra source of variation in the dataset. On top of the random variation within a group, we've now got some additional variation on top of that. And really it's that variation, the among group variation, that we're interested in. That could be the signature of something interesting going on in the dataset. So analysis of variance uses the principle that we have these 2 types of variation in order to ask the question, is the amount of variation we see among groups greater than we'd expect at random, given the variation that we see within groups? Because we know the variation we see within groups is the kind of random variation from one individual to another. If I collect a whole load of eggs and measure the amount of pyrazine in them, I'm going to see differences because of measuring errors, because of genuine small errors, small differences in concentration between individuals, maybe because of genetics or maybe because of something else like exactly how old the egg is or whatever. But that variation is not interesting to me in the context of the question, which is really, are the life stages different? So I'm going to say, given that individuals are variable, is the difference between the groups, the among groups variation, greater than I would expect at random? So just looking at that graphically, what would it look like if you were seeing not very much among group variation? It would look like this. So here we've got an illustration of a situation where there's relatively large amounts of within group variation. If we look at the green group there, we've got lots of spread for that bell curve. But the differences between the group means are pretty small. They're quite tightly clustered together. So in this situation, you look at that and you think, well, that could easily happen by chance under the null hypothesis that these 4 samples all come from the same underlying population. The opposite scenario is where each individual group or sample has a very narrow spread of variation, so relatively small within group variation, but instead we see big among group variation, nicely, clearly separated means. So in this situation, we think, well, this is pretty unlikely to have happened by chance. It probably therefore reflects a genuinely significant difference between the groups. Okay, so that is the basic principle of analysis of variance, and after the break we're going to look through quite a detailed worked example to help you get your head around how we do ANOVA mathematically. So that's going to be part 2, so I'll see you in a minute.